Just a second, when it is preparing to go live, as we go and we'll start. Yeah, go and check. All right, so I think we are live now. So good morning, everybody. Welcome to the Nitte University webinar series. It's part of the Consortium of Rare Genetic and Bone Marrow Disorders. We have these webinars on selected topics every 2 months. And today we have the resource person from industry background, and the topic chosen today for today's seminar is drug discovery and development, special reference to NCEs and biologics. We have the resource person, Dr. Ramchand, here. We also have with us Professor Dr. Satish Kumar Bhandari, the Vice Chancellor of the university, who is a clinician, an administrator, and most importantly, a researcher who has keen interest in following the developments in the field of research, particularly in biomedical research. Sir has been a great support for all the activities in the university, particularly for our institute, which is conceptualized mainly to carry out cutting-edge research, and the university has been providing us enormous support in terms of infrastructure, resources, and of course, in allowing us to do quite a few innovative kind of work. So before we start the webinar, I would like to welcome all the panelists. I also have our co-founder, Dr. Aarti Khanna-Gupta, with us. She is with me in this consortium. We set this together. And we also have Dr. Bharti Mittal. She is from, again, she is from an industry background. She is also associated with the consortium. She comes with a bioinformatics background. And both Dr. Aarti and Dr. Bharti, they were earlier part of the MedGenome company in Bangalore. So before we start the webinar, let me request our Vice Chancellor. Oops.
Hello.
You're back.
Hello.
Yeah, we can hear.
Hello, Anirban, am I audible?
Yes, sir, you are audible.
Yeah, it was muted for some time. I don't know. There is some problem. Okay, very good morning. And I think we are in the midst of a very unprecedented situation. Almost we are in the lockdown. Today we have got curfew in Mangalore. I think all over Karnataka we have got curfew. And most of us are working from home. And still the academics should continue. And I congratulate Anirban and colleagues for organizing this webinar in spite of all hardship. Very distinguished resource person, Dr. Ramchand, the president and CEO of Succin Life Sciences Private Limited, and Dr. Aarti Khanna-Gupta and Dr. Bharati, and all the distinguished participants in this very prestigious webinar conducted by our Consortium of Rare Genetic Disorders. Very good morning. It's my pleasure to be part of this webinar conducted by our NUCSER, that is the department designated for research as well as academics. And I understand it's a seminar series organized by the Consortium on Rare Genetic Disorder and Bone Marrow Disorders. And over the last 5 years, Nitte has made, our university has made very conscious efforts in promoting research culture at the university. And I'm extremely proud to say that recently, NAC has accredited our university the A+ category with an impressive CGPA of 3.4 out of 4. And really we are proud of this achievement. This score is a reflection of our collective efforts made by our teaching, non-teaching staff of the university, the constituent colleges, and particularly in teaching, research, administration, and community service. As I said earlier, NUCSER, the director of Anirban, is looking after our this particular institute, is one of the youngest institute in the Nitte family. And the main objective behind setting up this institute was to provide a platform to the faculty to carry out cutting-edge research in the field of biomedicine using state-of-the-art infrastructure and facilities. And I must say that the faculty of NUCSER have lived up to our expectations and have performed exceedingly well the last few years of existence of this prestigious institute. NUCSER has achieved phenomenal success and has contributed immensely in the research output of the university. It is evidenced from the large number of extramural funds obtained by the faculty, patent publications, and scientific communications in high-impact journals. The establishment of the consortium, this program is conducted by a specific entity of the NUCSER, the Consortium of Rare Genetic and Bone Marrow Disorders. This was started by the university with a seed grant under the policy of the university to promote research, particularly in one of the unique areas like rare genetic disorders. And this, I think, started about 3 years back, and it has done wonderful work in the last 3 years. I am pleased to know that Dr. Anirban Chakrabarti, who has co-founded with this consortium with the Dr. Arati Khanna Gupta, is committed towards bringing out very meaningful and impactful outcomes from the activities under this consortium. I'm told that the consortium has already embarked on a number of research projects aimed at delineating the genetic spectrum of rare, particularly mandibulofacial dysostosis and orthopedic syndromes and rare bone marrow conditions in our population. Besides being involved in research on rare genetic conditions, the consortium also conducts webinars on topics of relevance of today, and we have distinguished resource person from the industry, Dr. Ramachand, President and CEO of Succeed Life Sciences Private Limited. And I understand they got centers both in the country, in Chennai, and in the US. And it's a startup, I understand it's a startup biotech company operating from 2 countries. I'm sure Anirban will introduce Dr. Ramchand adequately to the audience after my talk. The topic that Ramchand is going to cover today is a very, very relevant one, according to me, particularly for our country. Although India is one of the largest drug makers of the world, number of drugs which has been manufactured, discovered rather, when we prepare a large quantity of drugs to support particularly the developing world, even the developed world. But discovered India are few. Although the topic of drug discovery and development is relevant for all fields of medical research, it's important particularly in this area of rare genetic research disorders. Since we got more than 300 million people the world over suffering from rare genetic disorders, and out of which almost 1/3 of the population living in India. I'm told that approximately 80% of these rare genetic disorders are genetic in origin and being monogenic and polygenic, and 95% of them do not have any FDA-approved specific modality of treatment. Thus, understanding the complex process of drug development is fundamentally is very important, and we have to develop strategies identifying potential targets that could become possible drugs for conditions where there is no prescribed modality of treatment. I hope I'm very hopeful that our learned resource person, Dr. Ramchand, who has years of experience in pharmacology, particularly in nanotechnology, will give you a very good overview of drug discovery and development from our Indian perspective. I believe the topic is very relevant for PhD students, research scholars, scientists, and clinicians, and it will be very extremely enriching for them. On behalf of the university, I extend a very warm welcome to once again to Dr. Ramchand and Arati and Bharati. And I congratulate NUCSER again, and particularly the Consortium of Rare Genetic Disorders for organizing this webinar on a relevant topic. Wish you all the best. May God bless you.
Thank you very much, sir, for your motivating words. We really appreciate you being here today, despite things so very difficult for the head of an university to be always to be concerned about all of us. So we really appreciate your presence, sir, and your words of motivation will definitely inspire us to do even better. Thanks for all the support, and thanks for all your cooperation and your encouragement. So over now, I would like to request Dr. Aarti Khanna to introduce our research person today, Dr. Ramchand. Over to you, Aarti.
Thanks, Anivan. Thanks a lot. So good morning, all. Thank you so much for being here with us. Despite the challenging conditions and circumstances, I hope that all of you are observing the COVID norms and keeping well. That is the most important part in this difficult time that our country is facing. So it gives me great pleasure. It is a great honor and privilege actually to introduce my mentor and friend, Dr. C.N. Ramchand, who is going to deliver today's webinar on the general topic of drug discovery and development, largely in India. I'm not paraphrasing the actual topic that he's going to cover. He'll cover that. I should warn you right at the beginning that this is a very complex subject, and Dr. Ramchand is actually going to give us only a brief overview of the topic. It's the proverbial tip of the iceberg, so to speak. This is a very, very intense and deep topic that will, in a short period of time cannot be covered and done justice to. So please bear with that, because this is going to be largely an overview. So Dr. Ramchand, as you might have gauged by what has been said so far, is a highly accomplished scientist and entrepreneur over the years. He is currently co-founder and CFO of MagGenome Technologies, which is based in Cochin. He's also CFO of co-founder of Saxon Life Sciences, which is based out of Chennai and San Francisco. And he's advisor to Genome Labs, which is based in Cochin, MedGenome Incorporated, which is based in Bangalore. MedGenome, by the way, is one of the premier genomics companies in India and is doing extremely well currently. He's also advisor to Sipwise Beverages in Chennai and Baroda. In addition to that, I don't know how he does this. He is also director and CEO of Theragen Biologics and director of many companies. I am not going to list them out because there are too many, basically. Several companies he's director of. In addition to that, from an academic standpoint, he is also visiting professor and PhD guide at Swinburne University in Australia, at University of Newcastle in UK, in Charotar University of Science and Technology in Gujarat, Slovak Academy of Sciences in Slovakia, to name just a few, and also Sumandeep Vidyapeeth in Gujarat. So previously, Dr. Ramchand was with Sun Pharmaceuticals. It's probably the largest pharmaceutical company in India. And there he was VP and head of drug discovery. It is there that he actually set up a multi-million-dollar drug discovery center, which was very successful in many ways. And I'm sure he'll tell you more about some of the work that was done there. He has received numerous awards and accolades, again, too numerous to enumerate here. And he has published over 70 papers and edited number of books also over the years. And the papers have been most of them are in international peer-reviewed journals, very well regarded. So it's highly accomplished in that aspect also. As you can see, as I said, he is a highly accomplished scientist and entrepreneur. And we are really very fortunate to have him here today with us to give us the talk. To me personally, Dr. Ramchand has been a shining example of a successful scientific career. This is all the opportunities that were given to him, he has taken them and done something with it. You know, many of us get opportunities in life which we use maybe partially and brush them aside. But here is an example of a person who has taken every opportunity he has got to its fullest capacity. This makes for a successful scientist, entrepreneur, and person. But above all, I feel that he is an amazing human being who has never shied away from helping everyone, uh, especially students. And as a result of that, he has mentored and taught dozens of MSc and PhD students who now themselves have successful careers. So again, in this short period of time, without taking up any much more time, what I'd like to say is thank you again, Dr. Ramchand, for being with us this morning. And I hand over uh, the virtual podium to you at this point. Thank you so much again for being here.
Thank you, Dr. Aarti, for wonderful words, and thanks a lot for organizing this program, especially to Vice Chancellor of the Nitte University, Professor Bhandari, and Dr. Anirban. And I have already told Aarti, which she has mentioned also, the topic what I'm going to talk, which is drug discovery and development, is a very, very complex subject. I also told that, probably it will take about 16 to 24 hours to complete as a workshop. I've taken these slides from about 350 slides just to give an overview. And probably very few things I can tell over that next 1 hour. So what I suggest is that you just do not worry too much about the slides, what I'm giving, but I will talk briefly what exactly is drug discovery and in the as simple way as possible so that students can understand that. You can see from the slides that multiple companies, these are various different companies, these successful startups. We started 5 or 6 years back. As Dr. Aarti mentioned, I was also in academics for a long time, subsequently shifted to industry. Then 5 or 6 years back, we started all these startups. Luckily, we are successful. Normally, startups is not easy to succeed, very difficult. For example, you can see that Saxon Life Sciences, one of the first molecule we developed and entered into phase 1 trial in US. We developed indigenously the entire thing, a Fab molecule. And probably if I get time, the second part of the talk will be how we have developed a live example, 100% from India. So we have various other companies. For example, MagGenome is a technology which we have developed here, a nanotechnology, magnetic nanoparticle, very successfully. And probably most of the Tamil Nadu and many of the states are using our technology for the RNA extraction. So far, about 3 million the RNA extraction kits we have sold. Also, we are developing viral transfer medium, molecular transfer medium, as well as RT-PCR kit, plus many other DNA, RNA, protein extraction kits. So this is a very good example of a work that is carried out in the universities, especially PhD program, and translated into a successful technology and entrepreneurship and commercialized. We have other companies called Theragen Life Sciences, which is an AND, abbreviated new drug application or generic molecule to be sold world over. So that is another company we have set up. Then Theragen Biologics is a similar company like Saxon Life Sciences. We are using Fab molecule, that is fraction antigen binding or part of the antibody for diabetic retinopathy. And Theragen Molecular Innovation is a novel company in collaboration with the US company, funding from US. We are using IgY, immunoglobulin Y, or chicken yolk derived antibodies for a specific antigen for multiple applications. One of the first applications we have started is on alcohol-induced hepatitis. So these are some of the companies successfully we have initiated and continuing. We have about 200, 250 people working altogether. MedGenome, I'm not directly involved, but I'm an advisor to that company. So is Cygenome. And as you know that Dr. Aarti was working in MedGenome. And let me also tell you, Dr. Aarti was a couple of years junior to me. She was one of the most brilliant students of our department. And normally that place, MS University, Baroda, it's very difficult to get it an admission. But once you get into, they go to very, very high level. That's why she was in Harvard University. Now, today I'm going to talk to you about drug discovery development, and I'll briefly talk about new chemical entities. What exactly small molecules or new chemical entities? What is biologics? Now, four different areas are there in this spectrum of product development, which is small molecules or NCEs, biologics, that is mostly recombinant proteins. The other one is medical devices and diagnostics. So somewhere or other, we are mainly involved in all these four different products. And my first experience was in NCE development, that was in Sun Pharmaceuticals. Now, since a lot of students are involved, I will briefly tell about what is a drug. Everyone knows what is a drug. But then there is very specifically something you need to understand. A drug is a substance that when absorbed into the body of a living organism alters normal bodily function. That is very, very specific. And these type of drugs normally acts on specific metabolic pathway, and in the metabolic pathway, specific protein. But most importantly, the drugs, a clear definition is that it has to be either for the treatment purpose or curing a disease or preventing a disease or a diagnostic purpose. So this is basically the explanation for a drug. And the drug source or NCE. Now I'm talking only about NCE, not about the biologics. NCEs can be from various sources, but right now the major source is synthetic or chemically synthesized. It can also from animal sources, plant sources, microorganisms. Now, let me briefly tell about drug discovery in India, drug discovery in the world. You all might know that there is very, very large number of diseases out there. Basically, there are 30 to 35,000 human diseases out there. And most of the disease, probably we do not know the mechanism. And since you are working on rare diseases, probably many of the rare diseases, mechanism is unknown. So 75% of the diseases still you do not know what are the different mechanism, or the target is yet to be identified. Basically, if you analyze and see 98% diseases, you do not have a target-based drug. This is the unfortunate situation. Now, if you see what are the types of diseases, like, you can have diseases like cardiovascular disease, because 1/3 of the people, they have cardiovascular disease, 1/3 die of cancer, and the remaining by other diseases like old age and infectious diseases. Now, if you see really cardiovascular disease, is there any drug? Really, I'm not going to explain, but it is really not identified a good drug. Cancer is coming up. Target-based drugs are coming up. On the other hand, there is very good vaccines are there. An example is COVID vaccine, very effective. Most of the vaccines identified is really working very well. So there is, what I wanted to tell is that still there is a great opportunity for discovering and developing drugs. Now, if you see out of all the diseases, central nervous system disease, like Alzheimer's disease, is there any drug? No. What about schizophrenia, depression, mania, manic depression? So there is a large number of diseases, still there is no drugs available. That means there is next 100 or 200 years, probably, we have great opportunity to discover, develop, and mainly the mechanism of the diseases, because that's the fundamental requirement to develop any drug. So first mechanism of the disease, after that the target, drug target, then subsequently the pharmacophore, and develop that one. Please do understand, it takes about 15 years to develop a drug. Now, probably you might be knowing that Professor Bhandari was mentioning that India is one of the largest producer, yes, 4th largest producer of any drug in the world, and we are the greatest exporters. But unfortunately, we have not developed any novel drugs. On the other hand, the drugs could be produced only by few countries, mainly US. All the top United States companies have produced most of the drugs. So only 7 countries in the world could develop um, novel drugs. Please also remember that the drug sales value, commercial value, or what is the Indian contribution? Roughly about 2,000 billion is the world sales value. 30% comes from the US. And whereas in India, it is only 1.5%. Our sales value comes to about 20 to 30 billion. So this is the situation. The reason is that in India, we do not have the patent value because we are all using generic molecule. No drug discovery has happened. If a novel drug, a worldwide novel drug is produced, that will take over the value chain of the whole 22,000 companies put together. So that is the value of research. Now, what are the types of drugs? See, there are 400 different classifications has been made. You would have heard about antipyretic, analgesic, antipsychotic, antibiotic, antiseptics. These type of 400 categories are there. That is for specifically the drug category. Please do understand only 500 basic drugs are there. And if you see about Me Too drugs, that means analoging that drugs, there are about 2,000 drugs are there. Whatever you are seeing is that lot of formulations are there. That means brands. So basically, the novel pharmacophore, as of now, only 400 to 450 drugs are there. Now, what are the different types of drugs? New chemical entity. What exactly is new chemical entity, which is otherwise called small molecule? Basically, these are which has got a patent or existing patent is there. What is generic? The moment the patent period is over, after 20 years or so, it becomes generic. So all over the world, companies get into that. Then the value of the original company's product also comes down. So India was extremely good in developing generics. Basically, we had extremely good process chemists, process and formulation chemists. Then comes biologics. There are not many biologics has developed, but recently in the last 10 years, many have come up. And it is expected that next 10 to 20 years, 60 to 80% of the drugs will be biologics in nature, like, interferon alpha, interferon gamma, erythropoietin, and mainly antibodies, vaccines, and drugs like that. For example, the drugs what we have developed, a Fab molecule or fractions of the antibody. So antibody going to be the major biologic part. So these are the things, and as I mentioned, few more part is there, which is generic molecules and other process, etc. Now, how the drugs and the drug discovery and development is happening. Now, you can see some slides, plus please do not worry about what I have written there. Again, to explain each and everything, it will take probably a couple of hours. So I'm just going to tell. Now, any drug development or discovery consists of few areas. The first part is drug discovery part. What is drug discovery? As I mentioned, the first part is that before discovering the drug, you need to understand the mechanism of disease. For example, how the cell proliferation takes place in a cancer, or in a cardiovascular disease, how heart attack takes place, how pain, for example, cyclooxygenase. And a typical example is that inflammatory diseases. The entire mechanism is known. How lipids are converted into through lipases, arachidonic acid, and how this is converted through cyclooxygenase pathway, the prostaglandin, leukotriene, and its receptors. Entire mechanism is there. Because entire mechanism is there, many of the drugs in the inflammatory pathway, pain pathway is known. So one is mechanism. Once after understanding the mechanism, you need to identify the real best target which do not affect the pathway and the human, the metabolic conditions. So once you identify that specific protein, there is a process by which to study that protein. You have to isolate, clone, express, isolate, purify, X-ray crystallographic structure, understand the structure, and where the binding. And what are these targets? These targets are normally enzymes, can be receptors, can be ionic channel, transduction protein, or transport protein. So understand this protein, study the X-ray crystallographic structure, understand the three-dimensional conformation of the protein, find out the binding pocket. So that part is over. Now the next step is that find out the pharmacophore. What is the type of molecule activate or inhibit that protein so that metabolic pathway can be inhibited or activated so that the disease can be controlled. So that is the second part. That means identifying the pharmacophore, and there is various mechanism by which this pharmacophore can be identified. So this two things comes under drug discovery, and that itself takes years and years. But then subsequent to that, the drug development. Now, 10 to 15 years or 17 years take the drug development part alone. That's a defined period that you will be able to do mainly preclinical studies, toxicological evaluation, and about 3 stages of clinical evaluation like phase 1, phase 2, and phase 3. So that takes about 10 to 12 years. And the cost of developing a drug, all these pathways, all these preclinical to toxicology to clinical evaluation, roughly about $2 billion. That is the type of cost, almost 15,000 crore. Please remember, no Indian companies will be able to manage up to this level. Most of the Indian companies who are trying drug discovery will go only up to phase 1 trial. But I am very sure in the next 10 years, many of the drugs is going to come from India too. Now, what are the different drug discovery programs, whether it is in India or internationally? So these are some few drug discovery programs I have mentioned. Process development. Indian pharmaceutical companies are extremely good in process development. Once you have the structure, once the drug is known, and if it is off patent, we know how to synthesize that using the existing patent methodology, what is prescribed, or you can develop your own pathway for developing that one. So most of the Indian pharma earlier made money through this process development. And out of 23 or 24,000 companies, 20,000 companies are involved in the formulation development. What is formulation development? Because once API, using the process, a drug is developed, it has to be formulated. That's what you are getting from the show, whether it is tablets or other oral drugs or injectables, the drug has to be formulated. Now, very little intellectual property can be given over there unless you are a drug delivery system. So the third thing what people are trying is the drug delivery system. That means a drug which is existing for oral, which can be given nasally or some other route, injectable. That is drug delivery system. Indian companies were till now concentrating on these 3 things: process development, formulation development, drug delivery system. But after the patent regime, the new patent like the GATT has introduced, then about 10 companies started the new drug discovery or small molecule development. There are 3 different ways it is done. Using non-target. If the target protein is known, elucidated, and the pharmacophore is known, it's comparatively simpler to improve the existing pharmacophore. So that is what is called the non-target and non-pharmacophore, and many of the companies are doing that one. Then there is another one called non-target but unknown pharmacophore. You know the mechanism, you know the target, but you do not have the pharmacophore. So there is a way by which the pharmacophore can be developed. That is using multiple technologies. You need to get the structure of the pharmacophore and develop that. Then a third category is that unknown target and non-pharmacophore. That means you do not know the mechanism. But it will not happen like that. But there are a lot of examples are there. A very good example is paracetamol. Until 12 years back, 15 years back, nobody knew how paracetamol was working. It was not on COX-1 or COX-2, but identified about 12 years back a novel target called COX-3. So that mechanism also is known. But until that time, if somebody has to develop a drug like paracetamol, it was little complex. But then that type of discovery is called unknown target and non-pharmacophore. Now, novel biologics, which I will briefly mention, how novel biologics, especially antibodies or fractions of antibodies, how it is developed. Then you can also develop novel biologics using studying the metabolic pathway. And another category of drug discovery is that alternate use of existing drug. That means an existing drug, you know the target, you know the pharmacophore, but can it be used for a different purpose? A typical example is aspirin, which was originally for pain or inflammatory condition, mainly cyclooxygenase pathway. Now later, it has been identified that aspirin at a lower dose by a totally different mechanism, that is leukotriene inhibition, can inhibit the platelet aggregation. So these are some of the drug discovery program which is used, whether it is in India, worldwide. As I mentioned, the Indian pharma is totally dependent on formulation development, and few companies are depending on process development, and very, very, very few companies are working on totally novel NCs, and virtually no one is working on novel biologics development, but most of the companies are working on biosimilars. In this background, we have developed that, as I mentioned, a novel molecule, FAB, for macular degeneration. So this is the current scenario which I mentioned. There is 30,000 diseases and development time is about 10 to 15 years time. And roughly currently that it has been estimated that the cost of R&D expense, including the complete development, it was about $1.5 to $2 billion. And total number of new molecules is absolutely small, but then this is significantly going to increase because we have novel method for identifying novel biologics and antibodies. So probably in 10, 20, 30 years time, most of the disease we might be having drugs. Now this slide simply tells the complexity of drug discovery, because you can't develop just like that drug. And if you are talking about small molecules, a very large number of molecules has to be synthesized to get one single drug successfully out. And these are the different requirement in order this molecule to screen and get out of the pathway and commercially successful. The first requirement is that you need to have a valid biomedical hypothesis. Without a valid biomedical hypothesis, unless you know the metabolism, unless you know the target, the FDA, nobody will venture to start a drug discovery program. Now, it is very essential that drug has to be targeted. I'm not going to explain because otherwise it is worthwhile explaining each of these points. Since there is no time, I'm not going to tell, but the drug has to be targeted, selective, and potent. Potent means that you can't give a 1 gram, 2 gram tablet 4 times a day, not possible. You can see in the last 20 years, the drug size has come from grams to milligram now. Physically stable, it has to be soluble. That means it should have a very good pharmacokinetics. It is to be permeable, whether it is cellular permeability or in the gastrointestinal tract, mucosal to serosal side. It has to be metabolically stable. The moment you take it, it is converted by cytochrome P450 into another category of the product. That's not a good drug. It should not induce other enzymes or receptors. That's not possible. The binding of the drug has to be reversible. A drug which is produced, it cannot go and bind to a receptor permanently. Durable. The molecule has to be non-teratogenic. You know the story behind the teratogenicity. Then non-mutagenic. The most important thing is the drug should be patentable. That means such structure, there should not be any prior art. If there is a prior art, such structures are known, you will not get a product patent. The company will not go ahead and developing such molecule. And finally, the most important thing is that you can't develop a small molecule with 20-step synthesis pathway because it will be unsuccessful. It is very important that 4 to 5 steps. That means the drug has to be easily manufacturable. So these are the conditions that needs to be adhered to. Then what is the drug discovery classical pathway? So if you see here, once you identify the disease, then it is very important that study the mechanism, understand the pathway, isolate the protein involved, then you study the structure of the protein, understand the way which it works, its sequence, then clone, express, crystallize, understand the binding pockets. The second part is that, you can see the structure is given. So you need to identify a structure which goes and binds to the protein, which is an enzyme inhibitor. That means the site where the substrate binds has to, or it should mimic the substrate. Subsequent to that, the screening process is there. Then toxicological evaluation, then go for IND, investigational new drug filing to FDA, or if it is in India, CDSCO. Now just see I just wanted to give an interesting the outlook on the journey of the drug discovery and development. Now, really any drugs, synthetic drug has got only last 100 years. Before that, nothing was there. Now, even the first 30 to 40 years, the drugs were developed by serendipity. That means accidental finding. There is ample examples are there. A typical example is penicillin itself. It was not a rational approach for developing penicillin, accidental finder. So there are two things. The development of the drug I have shown in the last 120 years, and the science, the development of various science development. For example, you can see the first, the development has taken place in chemistry. Subsequently, you can see microbiology and physiology came up. Then back in 1940, biochemistry came up, molecular biology in 1980. Then if you see really now, you have structural biology, computational biology, genomics, proteomics, transcriptomics, and synthetic biology. So this is the pathway, the science has grown. On the other hand, we have a very interesting drug from aspirin. Then subsequently molecules like insulin has come. At that time, 100 years back, insulin was extracted from animal. Now it is different. It is recombinant DNA technology, and there are a lot of improvements have taken place. So very interestingly, both side by side passed, and right now we have about 400, 500 different drugs for various conditions out there. Now, this is another interesting statistics have come. Probably at the peak of the drug discovery and development, 1995 or something, about 30, 40 molecules have come up in a year. You can see continuously it is declining because all the low-hanging fruits has been taken up. That means the one which is easy to develop has gone down. And it is expected that, by 2025, 2030, hardly any small molecules will come up. This is what is happening. You can also see worldwide, the number of small molecules is coming down. The expenses are going up, but the biologics is coming up, novel biologics. Now, what is a drug target? So, as I mentioned earlier, there are various drug targets are there. A DNA can be a target, a RNA can be a target, proteins can be a target. So these are the major targets. Probably the best target will be a DNA because 99% of diseases has some kind of SNPs or mutations are there. But it is very difficult to alter that DNA based on the current understanding because I'm not going to explain, but something interesting. The same way, RNA-based therapy, antisense therapy is going to be one of the best. Again, there is some problem. Only very limited success has taken place. Basically, right now, you are successful with all different types of functional proteins like enzyme receptors or ionic channel or transduction protein, translation protein. Now, once you identify this malfunctioning of that particular protein, then you need to work on that pathway. And that is the way, as I mentioned earlier, how we can modulate that malfunction and, the identified drug target, the drugs can be developed. Now, there are various possible drug targets, which I mentioned earlier. If you see that there is DNA, DNA is transcribed to RNA. DNA can be a drug, RNA can be a drug. Then this RNA is translated into a protein, and few of the proteins has become very good drug target. It is expected to have about 2,000 possible drug target. We have identified very well about 200 or drug targets. Now, another important thing you have to understand is that earlier drug target was based on isolated systems and working on isolated condition, isolated protein. But lately it is systems biology. That means there is an interaction between DNA, RNA, protein, and metabolites. So this is basically the molecular dynamics. There is a 20 billion opportunity for interacting one molecule into another. So right now, the drug discovery is looking into the systems biology, or just to understand that once system biology is known, you will be able to understand which all places the toxicity can take place. And apart from the target-based development of the drug, what are the other possible targets you can look into? Now, once the target is identified, there is a method by which target validation can be done. And after target validation, what are the things to be done? Now, knowingly, I have not given how the target is to be validated. It's a very complex subject. I was mentioning to Dr. Aditi that if you have any time after 2 or 3 months after pandemic is over, I'm very happy to come and give a complete workshop, which will take about 24 hours or 3 or 4 days will be required to complete this, the whole thing what I am talking is about. Now, what are the sources of potential drug candidate? Earlier I mentioned there are sources from microorganisms, sources from plants, synthetic drug, and there is a methodology called combinatorial chemistry. In combinatorial chemistry, once you have some idea about the structure or the pharmacophore structure, using combinatorially, you will be able to synthesize thousands, 10,000 or millions of compounds. So this is what is called combinatorial chemistry. So this is one of the successful way for identifying novel pharmacophores. The second is the substrate-based approach. That means if you know the structure of the substrate, which of course we all know, most of the once metabolic pathway is studied, identified, you know the target and the target substrate, you will be able to develop a drug based on the substrate structure. Then you can use natural products also, then microbial fermentation products, many plant extracts, animal extracts are used, insect extracts are used. So this is the way of various drug molecules are developed. But let me tell you, the job of a medicinal chemist in this category is very, very important. Once they know the structure, three-dimensional structure of the protein, based on the binding site, they will be able to synthesize the molecule. The next most important thing is that screening process. The drug need to be screened. If it's a combinatorial chemistry, millions of compound, then it has to be screened through a high throughput methodology. For example, if it's an enzyme inhibition assay, you need to develop a very robust method for the assay so that hundreds of compounds can be screened over a short period of time. The same cases with receptors or other transport proteins, transduction protein, ionic channels, etc. So this is a high throughput process whereby which a robust method is developed, and very quickly hundreds and hundreds of molecules can be screened, by which you will be able to get a hit molecule. I will be explaining that one. Okay. Now, once this assay is developed, so there are various types of assays are there. What we are talking is about the high throughput assays, always in vitro assay. Subsequent to that, a tissue-based assay has to be developed. It can be taking a tissue from an animal source or a human source, or it can be cell-based assays. So the second part is that, and the third is an in vivo assay. Invariably, FDA will ask for some kind of proof of concept in an animal model, whether it is working in an animal model. So, and not only that, every pharmaceutical company interested to know whether it is working in a animal model. Otherwise, that, later during the development, probably if it doesn't work, it's a problem. So these are the assays that at early stage needs to be developed. Now, one of the important thing is that all these assays has to be highly reliable and a robust assay has to be there. It should be easily, the feasibility is very, very important. That means all the resources for running the assay is required. Then automation is very important, such assays. If you are involved in screening thousands and thousands compounds. The second way is that there is a me-too development. That means an existing pharmacophore and improving that pharmacophore. You don't need to go for these type of high throughput assay in which that, single enzyme assays or, receptor assays can be used. Most of the Indian companies are currently improving the existing structure so that, the side effects can be reduced, the efficacy can be improved. Of course, the important thing is another cost. It should be a cost-effective method. Now, in the drug discovery process, there are drug development initial stage, there are 3 different types of pharmacophores are developed. The first one is hit molecule. Hit molecule means like, out of 10,000, you will get about 20 or 30 different hits will be there. That means the structure, the pharmacophore is sufficient enough to inhibit or activate the protein in question. For example, is an enzyme, inhibit the enzyme. So some structural requirements are there. Now, this will be further screened and, subsequently using the medicinal chemistry approach, you can improve the structure. And during that time, in silico methods can be used. That means using computer, the structures are available, protein structures are available in silico, in vitro, or in vivo. Improve the structure of the molecule to get a lead molecule. Once you get the lead molecule, that means that structure is fixed, fine. Then you have to test for its ability to cross in Caco2 cell lines or in the intestine, or whether it is easily degraded by cytochrome P450, or it has a toxicity mutation. Like that, a certain screening has to be done, and if it is successful, it will go for a drug molecule. And that drug molecule will be involved in the further development. So whatever you are seeing over here, this is the initial part. Normally, up to lead molecule, about 10 to 20 molecules will be developed as a lead molecule. And from that, 1 or 2 molecule will be developed as a drug molecule. So this, remember, hit molecules to lead molecule to a drug molecule. Now, after the initial development, that means all the preclinical studies, in vitro studies, or, ex vivo studies, animal studies, this has to go through a multiple toxicological evaluation. Before going through phase 3, there is more than about 50 different studies has to be done. But luckily for the phase 1, when you are submitting investigational new drug, there are not many toxicities there. You will have to do the Caco2 cell line study. That means the molecule is transported. You have to study the mutation. You have to study the LD50. That means whether the molecule kills the animal at a low dose or a high dose. It is very, very important that, it should be minimal toxicity or no toxicity. So about 2 species has to be tested, about repeat dose toxicity, LD50, subchronic toxicity, mutagenicity, etc. It can go for phase 1 trial. Then later, during phase 2 and phase 3, additional studies are there. For example, ADME study, pharmacokinetic study, then safety pharmacology. All these will be coming at a later period. Altogether, about 50, 60 different animal studies are there. So this is probably a recapping that, identify the disease. The first thing is that you need to identify the disease. Then identify and validate pharmaceutical target, which is the protein in nature, protein in question, and to which the drug has to work on. Then identify the lead molecule. That means the structure of the drug that needs to be developed, that the drug. Optimize lead molecule, get the structure, preclinical trials. So once this is done, and you have successfully completed the preclinical trials, then you can go for clinical evaluation. What exactly is clinical trial? So there are 4 different types of trial is there. In fact, 5 different types. There is something called phase 0 study also is introduced now. It was in the last 10 years. I'll briefly tell, I have not mentioned anything about phase 0 study. In phase 0 study, suppose you have to start with a particular amount in phase 1 study. Phase 1, what you do is that, there is something called MTD, maximum tolerated dose in animals. That means at that dose, animal will be having some side effects. 1/60th of that is the first human dose. That means, let's say that, the animal MTD study shows some side effect, let's say 100 milligrams per kg body weight. Then the humans can be 1/60th of that. Let's say about 0.5 or 0.8 milligram will be the first dose per kg. So if it is a 70 kg person, about 50 milligram can be the first dose to be given to human being. Now that is starting with phase 1. Then there is dose escalation is there, 1.5 to 2 or 2.5 times. So for example, starting with, let's say 50 milligram and perfectly fine in a healthy volunteer. Phase 1 always has to be done in healthy volunteers, except cancer and few other diseases. For example, any eye injectable disease has to be done in patients. Otherwise, it is in healthy volunteers. And in phase 0, 1/10 of the phase 1. Suppose, we decided that 50 milligram is the phase 1 dose. You can take 5 milligram to human, and with minimal tox studies, the FDA will approve that. This is to understand whether the drug can reach to the required places. For example, if we have to study something in the brain, does it reach the brain? It's very important to understand. So that's why this phase 0 also is introduced. So now there is a relationship between drug discovery and clinical trial. Clinical trial takes about 7 years to complete. Phase 1 to phase 2, phase 3, and there is one more, phase 4 also is there. So during the drug discovery process, all these assay development, compound design, all those things will be done. And many of the things work will be carried out thinking about how you are going to do the clinical evaluation. As I mentioned, it should be easily manufacturable. The dose should be as minimum as possible, etc. So there is a very clear cut at early stages only. You will have to decide what type of clinical evaluation you are going to do. Accordingly, you have to do the formulation, animal tests, etc. Now, this is the clinical trial process before starting phase 1 or phase 2. Again, it is a complex process, clinical evaluations. And let me tell you, it's unfortunate that India has not, I don't think any phase 1 trials has been done till date. Because invariably, most of the company, when I was involved in clinical trials, drug discovery in Sun Pharmaceutical, our earliest drug, we did not get the approval in India, went to Europe and get the approval. So recently also, many of the drugs, most of the company take to other countries. And one of the easiest place to do is Mexico, USA, Netherlands, Belgium, and Australia. So these are the companies, most of the people go, Indian companies. Then once you get the approval, phase 1, it can be brought back to India. You can go straight for a phase 2, but sometimes the regulator will tell again to do one more phase 1 trial in India. So these are the process by which a clinical trial has to be done. There is the hundreds of different people are involved. One of the major role is by the doctor, who is the principal investigator, and many other scientists are also involved. So there should be a sponsor, which is the company who developed the drug. Then protocol. No, invariably the company produces or develops the protocol and give it to the principal investigator. Site selection. Not every sites are approved by DCGI. So only approved hospitals you can go. Then there is an IRB, Institutional Review Board or Ethics Committee, a committee which constitutes about 12 to 14 different people, which includes doctors, scientists, a priest, an ordinary person, etc. And they have to approve the protocol. Then go for the regulatory approval, which is in India, it is DCGI for small molecule. and is RCGM, Review Committee of Genetic Manipulation for Biologics. In the US, only one regulatory body is there, but in India, there is 2 different bodies are there. But eventually, it will go to DCGA for the approval of the clinical valuation. Then patient screening is very important. You need to see the inclusion and exclusion criteria. Then patient recruitment. It's not very easy, especially in India, to get patients. It's a very tough task. Then subsequent that all the investigators has to be trained and certified. Then there is a clinical monitoring committee is there. They will monitor all the clinical trial process. Then site audit. Wherever you are doing the clinical evaluation, the site has to be audited and the data has to be managed and study close out. Then the report will be finally submitted to DCGI to approve to go for the next phase. That means phase 1 to phase 2 or phase 2 to phase 3. This is the process which is taking place. Now, what are the preclinical data required before going for a phase 1 trial? You need to understand the pharmacological profile of the drug. That means pharmacology, what is the target, how it binds, binding efficacy, association, dissociation constant, those type of all the pharmacological aspects has to be covered. There should be an acute toxicity of the drug in at least 2 species of drugs. Acute toxicity as well as repeat dose toxicity and short-term toxicity studies depending on the proposed duration of use of the substance is proposed clinical studies. For example, if you are doing a single dose in phase 1, only 1 dose has to be given and the study is only for a short time, then accordingly the tox study has to be done. For example, if a drug has to be given for asthma, which is a long-term thing, or somebody has to take allergic drug continuously, then the study, the tox study requirement, even at the initial stage will be, the duration will be high. The ADME study, administration, distribution, metabolism and excretion studies has to be done. And some efficacy studies. So these are the requirement, preclinical data requirement for a phase 1 trial. But when it goes to phase 2, additional requirements are there, especially connected to cytochrome P450, how the drug is metabolized and drug-drug interaction. There are multiple things out there. Now, this is briefly about the clinical trial. Now, I have just given that, at phase 1, how many patients are required or volunteers are required, phase 2, how many are required, and phase 3. So normally phase 3, many times it can be up to 5,000 to 10,000 patients also, but phase 1 normally is in the range of 20 to 100. But just understand that, 10,000 molecule, once it reaches phase 1, only 10% of the molecule which enters the phase 1 will successfully come out. So that is the unfortunate situation. Now there is one more trial, which is phase 4, in which that pharmaceutical company don't introduce this worldwide immediately, even after the approvals are there. What they do is that, a limited doctors, they introduce for a 1-year period just to understand whether the drug has got additional toxicological effect. Because initially, in a limited number of people, it is done a short term. So at least for a year, they will study, and that is what is called phase 4. And once that is successful, they will introduce the drug. Arti, the next set of, so do you have time? Because the time is really over. I think because the next one also will take at least 30 minutes. So I think probably I can finish here and ask questions. Otherwise, I can continue with how we have developed a biologic molecule.
Anil, can you take that?
Yes. Dr. Ramchand, if it is going to take 30 minutes, then I'm afraid maybe we'll have to request you to do it again some other time.
Correct. That's what
we can stop right here and take questions. I think if that is acceptable to you. Yeah.
It's easily 30 to 40 minutes it will take.
Yes.
I just wanted to tell you how we have developed in-house a novel molecule, a biologics molecule for macular degeneration. We're developing one more molecule, a Fab molecule, very interesting. The molecule that we have developed, probably the first time a novel molecule is developed, and there is something called RCGM approval also we got the first time.
Can you take one example, Dr. Ramchand? If we can go up to about, say, 10 minutes or so, can you take one example, if you can tell us quickly?
Right.
Or
Maybe
it takes-
briefly, maybe another 2-3 slides I will show. So this
Okay, yeah, that would be nice.
superior anti-VEGF antibody for the treatment of wet AMD. So
Okay.
you need to understand what is wet AMD, AMD. AMD is age-related macular dystrophy. Wet AMD is basically 15% of that age-related macular dystrophy will convert into wet. That is a neovascularization process. Lot of vasculature, lot of blood vessel will come in the eye, breaks down, and the blood comes out, and the vision will be lost. So this is our company where it is located. It is 2014. It is located, and a small but very good team developed this molecule, 100% in-house. The molecule using fast display was discovered in Toronto, University of Toronto. This is our company overview. Not going to go about this, too much about that one. And what are the highlights? Developed FAB 201. That's the name what we have given. It's a novel anti-VEGF, vascular endothelial growth factor. Vascular endothelial growth factor is a protein molecule. There are 125, 165, 161. So these are normally present in the eye. And wet AMD is a genetic abnormality. Probably after 60 years it comes, and losing the vision. And a very large number of people are there worldwide. Almost 250 million people are affected by AMD. Out of that, 15% become wet AMD, which is devastating. They will completely lose the eyesight. So about 30 to 40 million people are there, and 5 million people in India. Imagine 5 million people losing the vision. The problem is that current drug, Lucentis, which is costing about $2,000, $3,000, and about 8 injections are required. Imagine that. So our idea is that, bring down the price, develop a much better molecule, which we have done, and currently in the phase 1 trial. So this is our team. This is, it is very, very important when you are starting with a new company, you need to have world experts. For example, Dr. Sidhu is an expert in fast display technology. Jaykrishna Ambadi, I just met him US 4 or 5 days back. He is one of the most well-known ophthalmologists in the world. His brother Balakrishna Ambadi is the one who got at 16 years or 17 years MD from Harvard Medical School. So he is one of our advisors. The next company also is one of our advisors, is going to be the advisor. So basically, I just wanted to briefly tell about what exactly is wet AMD. So once it is generated, this is the way you are seeing that. Initially, you can see very well, but after later period, you will not be able to see. You can see as spots only. So this is right-hand side. You can see the full antibody structure, which is probably about 1,500 amino acids are there. 148 kDa is the molecular weight. On the other hand, this FAB molecule, one part, or the fraction antigen binding sites, that is what we have cloned expressed using fast display technology. And you can see the CDRs. So what we have done is that, we came out with a totally novel CDR. In the CDR, about 50, 60 amino acids are there. And all these amino acid sequence is something novel. That's why we are able to get an extremely high affinity FAB molecule against vascular endothelial growth factor. And this is the process of development. So first requirement is that, you get the right sequence, then synthesize the sequence. This is a synthetic biology process. Develop the clone, plasmid, and transfer into the clone, then express it. Then you need to develop a very good fermentation process. The most important thing is that analytical procedure, a very good analytical procedure to screen as well as understand the protein, and to understand the biochemical, biophysical characterization of the protein, and purification. Robust purification requirement is there because, the protein should be devoid of any other protein, host cell DNA, host cell protein, and endotoxin. And finally, formulate the problems with proteins are that it will denature immediately. You need to have a very good formulation so that, it will remain intact overall. So where this, with this slide, I will stop. So we have done all this process. That means fab generation, fab validation and characterization, fab production, in vitro and in vivo efficacy and pharmacology, proof of concept studies, scale up and regulatory approvals, toxicology, ADME and MTD, all this we have done. We have done GMP manufacturing, and we have done in India IND filing, and in the US we have filed. We have few questions, we are answering that. So where we are. So this is a novel molecule which we have developed. The details I am not going to give. Thank you very much.
Thank you, Dr. Ramchand. That was a very wonderful way of putting it across, very simple way of explaining the very complex process of drug development. If you don't mind, can you stop sharing your screen, Dr. Ramchand?
Right, yeah. Can you stop sharing? Stop sharing, yeah.
Yes.
Yeah.
So thank you very much. We have a couple of questions from the participants. I would like to put it across to you for your answers and clarifying the doubts. So one question.
Anirban,
Yes, sir.
Can I
Yes.
give some comments about?
Sure, sir. Sure. Please go ahead.
Am I audible?
Yes, sir.
Yes.
Very much.
Yeah. It was wonderful presentation by Dr. Ramchand, and I request him to, I think it was 45, 1 hour, 1 hour, 15 minutes, not sufficient. I request him to, if some other, after a week or so, the time permits, I request him to again address our researchers and students. And I request the pharmacy students also to participate in this talk. I don't know how many of them have joined. There's one request.
Yeah.
And as I think he said, after the COVID situation improves, he's willing to come to the campus. Sometime I again extend invitation to him to come to the university and spend 2-3 days or even a week here. I will be happy to host him here and have an interaction with him and my colleagues and my students. That's one thing. One more small query I'm having is regarding herbal medicine, because that's one area we got a lot of, I think, Western Ghats, we got a lot of herbal products, which has got potential to be very path-breaking drugs. But unfortunately, we are, because this is one of my experience here, I think from my village, I had identified one particular plant which has got very potential because it's being used by herbologists or native healers for treating cancer. Because I was interested and I brought it to the university, and we have done animal studies and cell culture also, and the tissue studies, cell culture studies have been done. And it has got, I found that it's very potential and as good as some of the anti-cancer drugs which are available in the market. But we are stuck in that thing. I request Dr. Ramchand to guide us how to go proceed, because we had some partner from Japan also. He also took the same extract and he said it is very good. It has got anti-cancer properties. But unfortunately, we're stuck in that level. Can you guide us how to go about it?
Experience in developing various nutraceutical products, herbal products into nutraceutical products. There is one issue with, you just see last 20 years, how many molecules, how many herbal products have come as a drug. Only if you see in the last 20 years, I know is EPA, eicosapentaenoic acid, fish oil, which is Lovaza, which is mainly for reducing triglyceride level. Now, curcumin, see, we have developed, this is a highly bioavailable curcumin. You can see this. we are going to introduce. This is for as an immune booster. Now, one of the product curcumin I developed is Curculife, which is going very strong in the US. Now, there is few issues are there. A curcumin is a typical example. Nobody could sort out the bioavailability of curcumin. This is for many of the natural product. This problem is there. That is one thing. So, of course, in the case of curcumin, you can see that it is highly soluble and is highly bioavailable. We have done in the humans also. Now, many other phytochemicals, this issue is there. One is that, and it is rapidly metabolized by cytochrome P450. But most importantly, this phytochemicals has to be extracted. Now, when you compare with extraction and synthesis, the extraction become very, very expensive. Now, if this can be synthesized, then it will become very easy. Now, the structures are very complex. So, what if someone is developing some of these molecules? I would suggest first introduce as a nutraceutical molecule. Simultaneously, go for the drug. We are exactly doing the similar thing for inflammatory IBD, inflammatory bowel disease, and CDAD, Clostridium difficile associated diarrhea. Basically, certain IgY molecule, we know that as a drug molecule is going to take time. So introduce as a nutraceutical. Nutraceutical complexity is much less. You can do using reverse pharmacology approach, clinical trial. So what I suggest is that the herbal product, what we have developed, instead of going as a drug molecule first, do straight away as a nutraceutical. You will get a proof of concept simultaneously, and which can be introduced very easily, especially in the US market. The easiest place for a nutraceutical to introduce in the world is US. There, you don't even have to, FDA will not question. You can just simply introduce them. In India, FSSA approvals are complex. So introduce in India, and 100 times more sales are in the US. Then after that, develop as a drug molecule from that, because that will take time. So you can make some money as a nutraceutical, and that money can be spent on developing as a drug molecule, and you will know whether it can be developed as a drug or not immediately. That's what my suggestion. That's what we have done. Everything. The curcumin I have done, because curcumin I have found that is a very good inhibitor of Psy kinase for allergic conjunctivitis, and it inhibits all the release of prostaglandin, leukotrienes, and histamine from mast cell. But the problem is that we went for the phase 1 trial. We did not get the approval till date. This I am telling about 4 or 5 years back story. But it can be introduced as a, as a herbal product, comparatively easy.
Thank you.
Thank you, Dr. Ranjan. Thank you, sir, for your comments and for the invitation that you have extended to Dr. Ramchand.
I'm very happy to come, and I can come with our team. Maybe two days will be fine. But the same lecture, probably 300 slides with more details. I was actually not satisfied the way which I have given the talk. We can't give in 30, 40 minutes, but it will really take time, because each slide can be explained properly, and especially students are there, they need to understand.
We will surely look forward to that. And as Vice Chancellor sir has mentioned, we have a very well-established pharmacy college, and I'm sure, sir, by the way, there are pharmacy students who have registered for this webinar today, and there are a couple of questions from the faculty as well. To Dr. Ramchand. So I'm sure we will definitely plan something once things become a little better in terms of mobility and in terms of movement between places. Sir, with your permission, can I go ahead with the questions? Yes. Thank you so much. So Dr. Ramchand, the first question is from Dr. Vasant Kumar. He's asking you, could you please comment on a present-day contribution of basic science research for novel drug discovery and demands for biological research and the funding allocations? So what would be your comment for that?
Yeah, without basic research, you can't develop, you can understand the mechanism. So if you really see the worldwide research in life sciences, everything will be eventually focusing on to understand the mechanisms of disease. Even if you are purifying an enzyme and study the kinetics, there is a reason to do that. That is to understand the enzyme's characteristics, where it is working. So eventually this will have some relation to a disease process. So it is so important to do the basic science to understand the mechanism. And if you see in the US, the maximum amount of money is spent next to weapons research for cancer research, and basically understanding the cancer metabolic pathway, and how this is leading to the disease. So it is very, very important basic study.
So we have one more question from Dr. Murli. He's asking you, is there any common mechanistic pathway for most of the diseases? And what is your take on mechanism-based research?
Right. Unfortunately, at this point, most of the diseases do not have the mechanism understood, though we have the complete pathway is very well known. But many diseases, a typical example I have given for inflammatory pathway. We know completely that in the inflammation, how it takes place, pain, how it is taking place, allergy, asthma, all these things, there is complete mechanism is known. But there are many, many diseases, still the mechanism is not known, especially if you, let's say Alzheimer's disease. In the last 20 years, 4 different drugs has been out, but all is withdrawn, because that means the real mechanism of Alzheimer's disease still not known, though there are 4 different novel targets has been identified, but it looks like that is not the actual target. So there are most diseases still we need to understand the mechanism, identify the targets.
So we have 2 more questions. Again, Dr. Vasant Kumar, he has another question for you. He's asking, is there any standard format for doses optimization when you're talking about cell-based therapy, like regenerative medicine in critical limb ischemia, or what would be the ideal template if you have to devise or if you have to determine a dosage optimization? Is there any standard format for that, or it varies depending on the kind of?
The first, this dose is established in animals, there is a conversion rate is there. For example, let's say per kg in animals, let's say rat is X amount per kg. So the humans will be something like 1/6 of that per kg body weight. So there is a standard way. If you are mice to human, I am telling, but that is not accepted by US FDA. So the actual dose is determined during the phase 2 trial. But phase 2 trial also, roughly they will decide a low dose, middle dose, and high dose. So whichever works, that will be continued. Then if you see a typical example is Dolo 650. Now paracetamol, what is a 40, 50-year-old, Tylenol, 40, 50-year-old drug, and initial dosage was 500 milligram or 1 gram. That was the dose. But about 15 years back, studied properly and found out the dose is 650 milligram for an adult. So the exact dose to identify, it will take some time. But this is exactly the first study in an efficacy model in animals. There's a method by which from animals to human, and that particular dosage will be used when you are initiating the phase 2 trial, not phase 1. Now, there are many conditions in phase 1, the efficacy also can be established, then it will become easy. For example, allergic condition, antihistaminics. During phase 1 in healthy volunteers, you will be able to find out a rough dosage that is required in phase 2 trial. So this is the way it is done.
Okay, we have one more question. This is from a student. She's asking, sir, can you please suggest some good labs for biotech fresh students, biotech fresh graduates, to learn more about drug discovery practical work?
I think we will welcome anybody. We have a program last 2 years we ran called B2SCP, Biotech Biopharma Skill Enhancement Program. This is funded by Kerala State Industrial Development Corporation. But normally we used to take from students from Kerala, 10 students and 10 students from Tamil Nadu or something. We trained them. We used to give them 15,000 rupees. And 1 year we really trained them. Not only various sciences, all the practical aspects, but mostly it is related to biotechnology. That means a cloning part, expression part, fermentation, analytical technique, formulation technology, purification, plus many other technologies. So that is one way, but because of COVID, that is stopped. We don't know when it will start. But in the meantime, some good students are there. We always take them, and if it is a B.Tech student, we give 6,000 students. It is a M.Tech, we normally give 10,000 just for a training. They don't have to, we will train them in different labs. We do that.
Okay. Do you take the normal MSc Biotech students as well? Or?
Yes, we normally, I mean, welcome to send the CV. We always need people.
Okay, so we actually at NUCSER in our institute, we run MSc Biotech as an postgraduate program, and we have quite a few students. So Garima, if you're listening, I think Dr. Ramchand has already answered your question about the possibility that is available, and I think you could, by the time you graduate and by the time, I'm hoping that things will improve.
Dr. Aarti can send the CV.
Yes.
Sure.
Right now,
Yes.
this new company we started, Trajan Molecular Innovations. So it's a very typical biotech company. We need few, 6-7 positions are open.
Okay, that is nice to know. So we'll be in touch with you, Dr. Ramchand. If there are good CVs, maybe we'll send it across to you.
Any more
Aarti, you
questions?
have you have a question? Yeah,
I have
quick.
two
Go
quick
ahead.
questions.
Go
One
ahead,
is
go ahead.
that in this time of COVID, everything has been very rapidly developed. I mean, it is even the vaccines have come out so fast that we are all still amazed at how fast it can. So that means it is possible to do it quickly. That is one.
The vaccine development is slightly different, I will tell you. If it is a drug, small molecule, it could never be done. Not possible. But in the case of vaccine, what exactly you are checking? You are injecting the antigen, which could be virus itself. The toxicology can be evaluated very quickly. One of the best advantage of biologics is that, you normally do not fail that much in a toxicological evaluation. Not like small molecules. Small molecules, 98% will fail. In biologics, it is not. Very rarely, the only is that when you are developing an antibody or something, anti-antibody development is the only problem. Normally, toxicology will not be there. So that is one part. The second part is that what you are looking, clinical efficacy is one thing, agree, but if sufficient antibody is produced in the body, which can be viral neutralization assay can be done. I was always thinking why to go ahead with this clinical trial itself. Because realistically, in order to do the clinical trial, you have to do in the trial about 100,000 people minimum has to be recruited. And in the US, I think Pfizer, they have recruited 250,000 people. Then they waited for 6 months, and who are the people in the control group and the experimental group, and how many people got COVID. If the experimental group did not less than certain percentage, then this vaccine is successful. But actually, that is not required. You inject and see the efficacy for a certain number of people, but you can do the antibody titration. Then introduce the drug. So as long as toxicity is not there, and I think that is what they have done, but still they have done a clinical evaluation. So it is possible in this case.
Yes. Okay.
Vaccines definitely is possible. Vaccine will not take that many years. Yes, that's an excellent question. Everybody has this thing in mind, how they could introduce this in 1 year.
Go fast.
But of course, see, let me also tell you one thing, everyone losing time where the regulatory compliance. When we got the C3 approval, it took almost 8 months to get the approval. First, they came with 52 questions, unwanted questions. And we had to answer over multiple meetings. So wasted time. So if you see, 5 years we have reached up to phase 1. In the 5 years, probably 2 years we lost through this regulatory submissions and compliance. So if they really want, they can do that.
So I have one last question, and that is about repurposing of drugs. So there are a lot of already developed drugs that are being given to patients for many different, especially in cancer. Now, if you want to repurpose the drug, do you have to go through the whole clinical trial again?
No. If you are a repurposed drug, as long as the existing dose or less than that, you can do with a single trial. That is efficacy trial, which is a phase 2/phase 3 trial. You don't need to do any tox studies. This is the advantage of drug repurposing. Only thing is that metabolic pathway analysis, where the drug, for example, a totally different target, you will have to show that one. That's a very, a typical example I mentioned about, aspirin. Aspirin, 2 different targets, which is proved, and the second dose, like, for platelet aggregation inhibition is a small quantity. If anybody is working on drug repurposing, is a wonderful area, because the valuation will be exactly the same if you have a patent for the novel application.
Okay.
Instead of 15 years, you can do everything in 1.5 years.
Super. Super. Yeah, thanks.
Thanks, Aarti. Dr. Ramchand, I think I have one more question for you, if you don't mind. So this is one question from one of my colleagues, Dr. Krishna Kumar. He's asking you, what is the view on issues related to adverse reactions and challenges in monitoring safety of herbal medicines, particularly in the Ayurveda context? So, what is your take on herbal medicines which are being used, and what do you think about the safety?
There are okay. It's a controversial question and controversial answer. I will tell you. First of all, when Ayurvedic medicines are developed, like, the people are taking the plant extracts from different area, and I have developed about, you might have come to the Vijayawada place, isn't it? So they are one of the pioneers in developing these products. And one of the things that we have in herbarium and the sources of various plant-based products. Now, if you are taking from Kashmir, the concentration will be one. If you are taking from Kerala, it will be another. If you are taking from Tamil Nadu, another thing. Sometimes many of the active ingredients, if you are taking from one place, it is not there. Who is going to check in Ayurvedic place? No, nobody is checking that. So that is one of the major problems. Now, apart from that climatic condition, it has been found that, at various climatic conditions, the way which the concentration of each ingredient varies. That is another thing. Now, traditionally, many of the Ayurvedic products, you can only use water extracts, and you can't use solvents. But many of the ingredients cannot be extracted without solvents like alcohol, methanol, acetone, etc. So I do not know. And who has done the real efficacy of these things? Otherwise, it would have come as a drug molecule. How many? These can come under botanical drug. There are not many things are there. There's one all related to gastrointestinal tract purification, evacuation, or for constipation. There are some drugs are there under botanical drug, which is in the US. In India, that category is not there. Now, the final thing is that I know personally that many people add other drugs. For example, they will put aspirin, they will put paracetamol, they will put ibuprofen. I'm not telling about very well known, for example, not companies like Kotak Ayurveda or something. Small people put this one, and many people use steroids. It's a well known thing. And it has been tested also and found out also. So I would be, very careful in taking any of the Ayurvedic medicine unless it is from a real good source.
So, Dr. Ramchand, questions are still coming in, but due to the paucity of time, we will have to stop in there. But before we conclude, as a scientist, I have one question for you. The concept of drug discovery has changed a little bit over the years. I think nowadays, from target-driven discovery to phenotype-driven discovery. And I think there are a lot of alternative model systems which are coming into the picture. I use one of them called zebrafish. So what is your take on use of these alternative model systems, so which the non-classical model systems, particularly in small molecule screen? And how do you think the phenotype-driven drug discovery, is it better than target-driven? Or
no, even if it is phenotype, because your drug molecule pharmacophore is no way going to act directly on the any phenotypic change. It has to be target, has to be there. It has to be target, which is covered by the cell, which is the tissue, which is eventually the phenotype. So it is very, very important the target protein, because all assays are developed based on that. Now the zebrafish, of course, is a very good model that everybody approves because accepts, because many of the protein structures and sequences is something similar to whatever you require. So as long as that is there, it can be used. Definitely it can be used. And it is using. In fact, Lalli Ramakrishnan for the TB, she's only zebra model she's using in Cambridge.
Yes.
So
Of course, that's I think they're using Mycobacterium marinum, I guess.
Yes.
Because it's the natural host for zebrafish.
Yes.
I mean, zebrafish is the natural host. So I think we will conclude here. Thank you so much, Dr. Ramchand, for taking your time out. I understand you have just returned from the US and the time lag and the jet lag and all of that. In spite of all of that.
Serious. Yesterday I could not sleep. I was really worried to give this talk. I thought that, my cognitive function was not proper.
But so kind of you. Thank you very much for that commitment and for accepting our request and for being here today. And I'm sure it was a very interactive discussion. And our Vice Chancellor sir is going to ask me on Monday whether I will do something more about it. And I'm sure we will bother you depending on your schedule.
You're happy to come and give a workshop anytime.
Yes, we will take that forward. We'll take that forward very much.
If there is a talk to be given, it is better to give drug discovery a small area, but this can be explained properly in full. This was a very, very, kind of an overview only.
Now, I mean, this is just the curtain raiser, I guess, and we need to, make good use of this new connection that you have developed. Thanks to Dr. Aarti for introducing you to us and for, allowing us to host this webinar. Yes. So thank you so much for your time. We can only thank you and we can only appreciate your interest and your commitment to this webinar. Thank you very much. I also thank Dr. Aarti for facilitating this webinar through your contact with Dr. Ramchand. And as you rightly mentioned, it reflects quite clearly how good a mentor he is. And I'm sure he will be able to help some of our fresh graduates as well. We will probably, from the institute perspective, we will probably put that across to you. And if you find the CV suitable, Dr. Ramchand, I'm sure he will help us. Yeah. I would also like to thank our Vice Chancellor, sir. He has been very supportive of all these activities. And the fact that he has extended an invitation to Dr. Ramchand, it clearly shows his interest in this field. And I thank him for joining us today. I also thank other members of the consortium, Dr. Bharti and other colleagues who have joined. I have to thank my IT team, Dr. Mahesh and his team, for facilitating it on Zoom. Generally, we do it on Google Meet, but because today is a curfew, we are doing it on Zoom because from the institute, we stream it live, but because we are not allowed to go to the institute today, so we are doing it on Zoom. So thanks, Mr. Mahesh, for all the help. And I also thank our staff development cell, Mrs. Deshmukh and her team for helping us with the registration and all of that pertaining to this webinar. So for all the participants who are still online, I think you will have to fill up that feedback form to get your certificate. The feedback form is posted in the chat box. You can click on that and get into the feedback and click on that, and you will be able to receive your certificate for participating in this webinar. So thank you once again, everyone, particularly Dr. Ramchand, for your time and for giving this wonderful overview on the drug development, and for thanks in advance, I would say, for all that you'll do for Nitte and for this consortium. We look forward to your support and your cooperation. Thank you once again.
Thank you, Dr. Aarti and all the team, Dr. Anirban, Professor Bhandari, Dr. Bharti, and all of your team. Thanks a lot. Thank you very much for giving this opportunity. And as we discussed, We're very happy to come and give a workshop anytime.
Yes, sir.
Once the
We will
course-
take it. We will take it forward. Yes. Thank you very much. Thanks a lot. Yeah. Thank you. So with this, we end our webinar today. Thank you once again.